I will talk about a vaccine production we did, or a vaccine development we did, and I will go through that, but in between I will guide you on the way what happened all in between and our knowledge, and in the end I will raise some questions which have over the years, not just in this last 2.5 years, come up. Okay, skipping right through the first lot. And we'll go into the disease itself. I'm talking about Actinobacillus pleuropneumonia. It causes respiratory disease. It's a disease called pleuropneumonia in pigs. It's a devastating disease. The pigs actually grasp for air, and they die a horrible death, basically. And it affects pigs when they're in the age to 8 to 22 weeks. In pigs' term, that is an age where they nearly go to market. They go to market, to the slaughterhouse at 22 weeks. So it's in the last stage of their growth. Hence, it's a huge economic impact if you get pigs dying at this age. So, major disease for the industry. Problem, the vaccines that are currently there are not very effective. Some vaccines they actually have to give 4 or 5 times to have some kind of effect. So what do we know so far? We know that there are 15 seroviruses. Some of them are pathogenic. Others, some are highly pathogenic. Others are only mildly pathogenic. And when we've done some pure infections, Serovirus 5, which is a highly pathogenic one. So this is basically challenging the pigs with only their disease itself, with Actinobacillus pleuropneumonia. Nothing else is happening in the pig. We see that the pigs are dying within 5 days, or they had to be euthanized. They're basically displaying the symptoms I just said. They're grasping for air, they can't breathe, they have a high temperature throughout. Serovirus 15, it's in challenge experiments, it's only mildly pathogenic. The pigs didn't have to be euthanized after 7 days after challenging them with a high dose. They have high fever, they are certainly lethargic, but they're not dying and they're still running around. These are some lungs we have on the top here, Serovirus 15. It still gives us some pretty strong lesions. This is a lesion, typical lesion of Actinobacillus pleuropneumonia. This is Serovirus 5, definitely much more pronounced, the disease. So what we have to take home from this is that Serovirus 15, which I will talk about later on, there are some pigs that weren't even affected, and certainly none of them had to be euthanized. This is a disease by itself. We know that the disease is caused by the toxins. There are 3 toxins that are responsible for this. There's the APX1, APX2, and 3. You can inject the toxin by itself. They will actually cause the disease. There is a different combination of these toxins in the serovirus. We have the APX1 is a very strongly hemolytic and strongly cytotoxic toxin. It occurs in Serovirus 1 and 5 and some others, but I'm focusing the blue ones. These are the ones we're focusing on because these are the main ones in Australia, the main serovirus we have. We have an APX2, which is in most of the serovirus, except in Serovirus 3, 10, and 14. Amongst this group is Serovirus 7 that has only this toxin. Then we have a non-hemolytic but strongly cytotoxic APX3, and that is in a lot of serovirus, including Serovirus 15. So we know, so these are the main serovirus we have in Australia. There is Serovirus 1 and 5, which are highly pathogenic. They have the toxin APX1 and APX2. There's Serovirus 7, moderately pathogenic, has the APX2 toxin, and then there is Serovirus 15, also moderately pathogenic. It has APX2 and APX3. So that was the knowledge. There was also some research done with streptomycin-dependent mutant, and it for CR1, and there was an indication that that worked very well. So we were asked by the funding body, can we produce a streptomycin-dependent vaccine? So streptomycin basically means that the vaccine, if a strain is streptomycin dependent, it means it can only grow in the presence of streptomycin. So what happens in the pig, there's either little or no growth, and hence it is not pathogenic to the pig. So we created mutants for all the common CRVAs in Australia, which is the 1, 5, 7, and 15, and we did safety trials. So this is where I will leave the vaccine off for a while and tell you what has happened in the meantime while we're producing this mutant. We started to get some new knowledge. We did a study on pleurisy. And this is pleurisy in an abattoir. We wanted to see what species of bacteria or what virus we could find in pigs that had pleurisy at an abattoir. These are pigs that walk into the abattoir, healthy pigs. Pleurisy is, what normally happens is that the lung attaches to the pleural cavity, and this is a big problem because we actually have to rip the lung off, and it slows down the whole process in the abattoir. So it's a big problem. So we decided, yeah, we'll do that. We'll have a look. So it's basically pleurisy. Chronic pleurisy means that the pig is not growing very well. For the farmer, it means that they cannot send the pig or have to withhold the pig for 8 to 12 days till it actually reaches a weight so they can send it to the abattoir. So it's a huge cost for them. At the abattoir, you get waste, carcass waste, and obviously the whole line slows down. So we looked, every second week we went to the abattoir and we collected lungs. We collected a total of 210 lungs from 46 farms. And there were mainly 5 lungs per farm. And this is all the bacteria we found. So it's a little bit overwhelming. So these are the bacteria. This one here is the farm, and how many farms we found them. Now, we have, I highlighted in green, their primary agent, Mycoplasma pneumonia and PCV2. They also very known for being synergistic. What that means is, for example, PCV2, it suppresses the immune system, which makes, and both of these by itself are not very pathogenic, but because they suppress the immune system and other pathogens can come in, that means they're causing lots of problems. Then we have the primary pathogens, which we always also found. Now the story to take home from this one is that there was a lot of farms that had either Mycoplasma pneumonia and a lot of farms that had PCV2. This is out of 46 farms. So that is a problem in itself. The next one are all these guys in red. They are all secondary pathogens. That means they're really opportunistic. If there's something going on, they can come in. So they're normally not regarded by themselves as a problem. But I found it amazing that at an abattoir, where the pigs are healthy, you find all these pathogens. So basically, from that, we learned, well, there's a lot of secondary pathogens, especially Streptococcus suis and Pasteurella multicida were in large numbers. And we had the synergistic pathogens in large numbers as well. So that's the background. We're calling this porcine respiratory disease complex. And the reason we're doing this is because there are multiple agents or reasons why a pig comes down with pneumonia and why it has failure to gain weight. So the agents are among them, which we just listed, the primary pathogens, which can infect by itself, and then there's the opportunistic pathogens, the secondary. Sometimes an opportunistic can also be a primary, or vice versa. But then there's lots of other factors that could potentially help or be synergistic in a sense, or secondary. There's factors such as parasites. There's the environmental stresses, such as overcrowding, cold spells, other diseases, or underlying conditions. Anything that stresses the immune system. So we then decided to have a closer look at the toxins. The reason we did this, there was a vet and he told me, I have silver 15, but on all my farms it seems to display differently. On some of it's highly pathogenic, on others it isn't. Well, that of course could be some of the underlying other pathogens. I mean, that's farm dependent. But we thought, well, we better check the toxin profile, whether that is actually the way it's supposed to be. So we know from that toxins are encoded in operants, the contiguous genes. So we have the A and C. The A is the structural protein, toxin. C encodes the protein that activates the toxin. B and D are proteins that are involved in secretion of the toxin. So when you look at this, and I will only look at the main ones, so we have silver 1, for example. To have both these, they need to have the operants C, A, B, and D of the APX1, and they also need to have the APX2, C, and A. For some reason, APX2 toxin pinches the APX operand BD of the of APX1. And then we have silver 15. It has to have these operants again pinching the operand of APX1 for the APX2, and then it has to have the operand for the APX3. So we did another study, and we also, while we were at it, we looked at the genetic diversity. We used the enterotoxic repetitive intergenic consensus to have a look there. And we looked with the PCR at these genes. So what we found, first of all, the diversity. I probably should go one step back. Silver 5 was also always believed to be clonal, while according to this, we found 15 different patterns in the silver 5, which is a 38% diversity. It's not. We found also, especially in the silver 1, huge diversity, 61% diversity. So there is certainly some diversity within each silver, which quite surprised us. Then we looked at the toxin, the genes, and with the stars, that means that the stars means that this is the profile we expected, and that's the profile we got for most of them, but there are exceptions. This silver 1 didn't have the APX1CA. That means that's the one that displays the toxin. So what we did, because silver 1 is the toxin is strongly hemolytic, we put it onto a blood plate, and it does not display hemolysis. So obviously something has happened with this toxin. Silver 5 was normal. They all fell within what we expected. Silver 7 also had one, and it should theoretically, because it's missing the APX1 BD, it should not display any toxin whatsoever. We plated that out as well because APX2 is mildly hemolytic, but there was no hemolysis seen. And then we had 17 of the silver 15 that were quite different. And they should only display APX3. APX3 is non-hemolytic, but it should have had the APX2, which is missing. So it should not be hemolytic, and plating it out, it wasn't. So there's certainly something going on. And that's basically the summary. I just told you, skip through that. It's got more complicated instead of easier. We had, in the meantime, a lot of species sent to us, a lot of isolates sent to us that we couldn't identify. They were somewhere in the Actobacillus species, and we didn't know what to do with them. We knew that the 16S couldn't differentiate them, so we decided, well, we better have a look at these. So we did a multi-locus sequences analysis. We used 3 genes that have previously been proven to predict whole genome DNA similarity, and that's the REC-N, RPOA, and THDF. So we sequenced them for 36 isolates. We had 36 isolates, and we looked at the genomic similarity index. And we also did a species-specific PCR for Haemophilus pyosuus. We actually did 2 just to exclude this species, make sure that it is not that. And here are the trees. At the moment, we're still working on this. So at the moment, these are 3 trees for each gene. But what you can see in green here, we have a new species. And it's very closely related to Indolicus, but its genome similarity index is 0.56. So basically what we now have, to complicate things even more, is a new species. And this species, the vets have sent us in the pathology report and pictures, and it is very similar to Actobacillus pleuropneumonia. So this will confuse everything even further. So we have potentially one, there might even be potentially more. So now, just to sum this all up before we go further, so we have synergistic pathogens, lots of secondary pathogens, new species, diversity within the A. pleuropneumonia serovars, exceptions to toxin profiles. We have porcine respiratory disease complex, so other things could add to this, and other causes by the pathogens we're actually looking at. So then it was finally, with all this happening, it was in the last month we actually went and did a vaccine efficacy on silver 15. Now, remember I told you before that silver 15 is moderately pathogenic. We can't make this any in a trial setting. We can't make this really pathogenic, like not only moderately pathogenic. However, on the farm where we worked on, this is a major problem, and the pigs are dying of it. And the vet kept on saying, it's not moderately pathogenic, it is highly pathogenic. But I said, no, it's not. So we did the trial there. We got some pigs from their free-range farm, which never had an outbreak of A. pleuropneumonia. And we tested them with serology, and they were certainly all naive. So our setup was we basically had 5 animals, control animals, and nothing happened to them. And then we had intramuscular vaccinated pigs, 8 of them, and intranasally vaccinated pigs, also 8 of them. They were vaccinated twice. So we vaccinated them at 3 weeks with our streptomycin mutant, which came from that farm. So the strain came from the farm, we mutated it, and it's streptomycin dependent. 3 weeks vaccinated at once, second vaccination at 6 weeks. We left them alone for 5 weeks. 11 weeks we challenged them, and then we had them for a week, monitored them, and then they were all euthanized after the challenge. So this is what happened. This is the outcome. And hang on, I think I marked this. Okay. So basically what happens here, we can see the lung score. This is the control group. This is the intramuscular, intranasal. You can see that there's definitely more pigs that had a very low lung score, and the lung score of the intranasal was a little bit higher, but compared to the control group, there was a difference. There was definitely a difference in the lesions, especially in the intramuscular. When you look up here, they had 4 lesions. So there was, and we found APP in all these lesions as well. When you look at the body condition, I wonder if that goes. What have I done now? Okay, great. Oh yeah, must have a slide in there. That's okay. Thank you, Pat. And so then we looked at the weight. There was definitely there was something happening here. Overall, they lost a lot of weight. You could actually look at these pigs and say, yeah, they're very tucked in. They're not eating properly. This didn't happen in the other groups. When we go on further, so this is the observations we did. The first row here is at how many observation points did they actually display symptoms. So we had 41 times where we actually looked at these pigs. And you can see here that certainly the control pigs had lots more clinical signs, at lots more points, they had clinical signs. The highest score, well, we had one pig here that had an 8, so that threw everything out a bit, but in the intranasal, the score was definitely lower. The day they first showed signs, and even if that was only nasal discharge, was certainly much earlier. Here, I mean, one pig showed it at 6 days, which is 1 day off a necropsy day. Certainly, there was a difference. So if we would do a vaccine trial, we would say, yeah, this is working. Here, these are the lungs. You can see huge lesions of the control pig. These are the lungs of the intranasal. The only one that was disturbing, so most of them looked normal. This has one lesion, but it's on this side, but the picture was really bad on the other side, so I put this one up. It's only a very small lesion on the other side. This one has a big lesion. That was a little bit disturbing. And then this is intranasal. And you can see here, certainly the lesions are not as big. They are smaller lesions. Now, having said that, when we do vaccine trials, a vaccine will stop the pigs from dying. It will stop major clinical signs, but it will not prevent all the lesions. So we don't expect a vaccine to work 100%. Now, this trial was done with, this is the 5 control pigs in a shed. We had to keep the control pigs in a shed by itself while we were vaccinating the others, so just to prevent any, because one was intranasal, just to keep them apart. When we had them all vaccinated, they came together with the others. But these little pigs have a marvelous time in a huge shed, which is meant for 200 pigs. So they had a marvelous time. They were wonderful. This is treatment groups, and later on the vaccine, the control group came into the shed as well, and they had a shed, the same space. This is a pen that normally I think would hold up to anything around 30 to 50 pigs, depending how much they stock. Well, there we have our little 8 pigs. So they were not stressed. They were quite comfortable. They were not overcrowded. They were not fighting for food. There was heaps of fresh air. There was not much ammonia around. There were no other pigs that could possibly infect them with something else. Mind you, despite that, we lost 3 pigs, 1 pig per group due to other issues. But they were relatively nicely kept. And that's the same with all our trials we ever do. I mean, we do them at CAS. You can't even stress the buggers. They're just happy. So, and this is now where it comes. We now have to decide, will this vaccine work? if we go into the normal pig shed, where it's normally overcrowded, the management is not really wide, is there going to be protection? Is this protection we're seeing here, is this going to be enough? Will it work in a non-ideal environment? Can it work in the presence of other pathogens? Can it work with prolonged challenge? Can it work when with stress, suppressing the immune system? So basically, the point I'm trying to make is, why are we addressing such issues that depend on that everything is right? Because a vaccine depends on that your immune system is working wonderfully, when we know that the basics are not right. So is it sensible to focus on only one thing without addressing all the other problems? It's a general consensus that if proper management is in place, such as no mixing of ages, good ventilation, managed temperature, good hygiene, no overcrowding, good biosecurity, then most of these disease issues are actually not a problem. And then we have the antibiotics, the major drama as well. So research has only come through and just says that by treating at an early age with an antimicrobial, we're compromising the immune system. Studies mainly done on salmonella in mice have shown that antibiotic administration can paradoxically weaken immune memory, leaving a recovered host fully susceptible to reinfection with the same pathogen. Studies in pigs have suggested that a higher microbial diversity in the intestine in adult life is accompanied by more active genes involved in the immunity. So coming back to the problem, there are a lot of secondary pathogens that we have discovered. We have discovered new species with similar symptoms. So will attacking everything with a vaccine solve the problem? Will other species evolve to take the niche if you get rid of one species? I personally think that we need a holistic approach. We need to look at the whole system instead of just part of the system. I'm not having all the answers here. I'm not coming here and telling you this is the answers. I don't have them. I'm just wondering. We have attacked the problem with several ways in this research. We have looked, well, not only at the vaccine, but we have looked at also at antimicrobial resistance, which I haven't covered. But we have looked at that. We have looked at new species. We have looked at other species involved. We have looked at the genetic diversity of the serovirus. And we're looking at the diversity of the toxin. But I realized that this vaccine, without addressing all the other issues, will most likely fail. We need to have some kind of system approach. Somehow, disease needs to be seen as a failure of the system rather than just focusing on one facet of it. And that's basically where I'm heading. I don't think we're tackling this right. And with these thoughts, I'll leave you. I'd like to thank all my team. They're just wonderful. We've done a lot of work in the last 2.5 years, and they all have absolutely chipped in and done a wonderful job. And I'd like to thank all our collaborators in all the projects. And I leave you. This was where we stayed in the caravan doing night shift in the pig shed. And that's what we woke up to. Thank you.